Now, I want you to remember the activity we did with the bean soup factory. And if you think about what the nucleus did, or what the DNA did, its job was to copy the original recipe for the bean soup and pass that copy that they made on to the ribosomes. And so if you think about it, DNA is sort of the original recipe that your cells have for building proteins. And RNA would be sort of like that copy that was made. So in the process of making proteins, RNA is the copy of the recipe that gets handed off to the ribosomes. So to remember that, out to the side of DNA, let's go ahead and write original. And out to the side of RNA, let's go ahead and write copy. So you can remember DNA is the original recipe, RNA is the copy that gets passed to the ribosomes. If we go over to the first part of your table, there are some differences between DNA and RNA. The first difference we're going to look at is the type of sugar that they have in them. The sugar that DNA has is called deoxyribose. And the type of sugar that RNA has is called ribose. Both DNA and RNA are made of nucleotides, and so a nucleotide, remember, has a sugar, a phosphate, and a nitrogen base. Well, RNA has these too, but the sugar that they use for their nucleotides is not deoxyribose, it's ribose instead. Now we're going to look at the location where they're found. DNA is going to always be found in the nucleus. It can't leave the nucleus. However, RNA is made in the nucleus. Remember, it's the copy that gets made in the nucleus. But it moves to the cytoplasm after. And it has to move to the cytoplasm to be able to get to the ribosomes. The third difference between the two, DNA and RNA, is the bases that they have. DNA has the bases adenine, thymine, cytosine, and guanine, but RNA has the bases adenine, thymine, it does not have thymine. It has adenine, uracil, which we're going to use a U, cytosine, and guanine. So this is uracil. That's the one that's different. Uracil replaces thymine in RNA. And the last difference is the number of strands that they have. DNA is a double helix, so it has 2 strands, but RNA is just half of that. It has only 1 strand. Now we're going to look at some different types of RNA. If we come down here, there are 3 types of RNA. The first is mRNA, which the M stands for messenger. It's messenger RNA. tRNA, the T stands for transfer. And we have rRNA. The R stands for ribosomal. And messenger RNA is made in the nucleus. tRNA is found in the cytoplasm. And rRNA is in the ribosomes. Now we can look at these pictures of the two sugars in RNA and DNA. We've got deoxyribose here. If you notice, it's missing an oxygen where RNA has an oxygen. So this is the difference between the sugars in DNA and the sugar in RNA. The reason that we call the DNA sugar deoxyribose is because it literally means without oxygen. And so the meaning of deoxyribose is without one oxygen, because it's missing that oxygen that ribose has. Now we're going to come down here, and our job is to identify which of these nucleotides belong in DNA and which belong in RNA. In our first nucleotide we have R for ribose. That tells us that this is a nucleotide found in RNA. And it has a base letter A with it, and if we go back up to our table, RNA does have adenine as a base. So that tells us that this is a good nucleotide, and so we're going to put a square around it, because our job is to put a square around the nucleotides that can be found in RNA. Now we're going to come down to the next one. We have an R for ribose and a T for thymine. So that tells us it's an RNA, but T is not a base that can be found in RNA. If you look back up at your table, T is not there. We have a U instead. So this nucleotide does not exist. Our next one has a D for deoxyribose, so that tells us it's in DNA, and it's paired with A, which is a good base. It's found in DNA. So we're going to put a circle around this one, because our job is to put a circle around the ones that belong in DNA. And then if we move on over, we've got a D for deoxyribose, so this is DNA, paired with a U, which is uracil. Uracil is found in RNA, so this is not a good nucleotide. Then we have an R for ribose, which is found in RNA, paired with a G for guanine. This is a good nucleotide because G is found in RNA. Then we have an R for RNA with a U for uracil, and uracil is found in RNA, so we're going to put a square around that one too.